today we will discuss the range of the DNA manipulated enzyme, right? So there are several commonly used DNA manipulated enzyme in the laboratory. So for example, the nuclease can cut, shorten, or degrade the nucleic acid molecule, and ligase and join the nucleic acid molecule together. Polymerase that make copies of molecule, modifying enzyme, remove or add a chemical group, and topoisomerase introduce or remove the supercoil from the covalently closed circular DNA, right? And for the nuclease, so there are two different types of nuclease. One type is the exonuclease. Exonuclease basically it's like here, they can remove the nucleotide, all right, one at a time from the end of a DNA molecule. That's the exonuclease. And then endonuclease basically can break the internal phosphodiester bond, right? So here you can see this sugar phosphate, and then this is the phosphodiester bond, right? And then they can cleave the phosphodiester bond internally within the DNA molecule. That's the nuclease. And then for the exonuclease, right? So there are two different types of exonuclease. For this one here, there's about 31. So basically, they can remove the nucleotide from both 5' end and 3' end, so that you can see. So here is the top strand 5 to 3, bottom 5 to 3, and then they can remove from both 5' end and 3' end at the same time. Okay, so that's the about 31. So you can remove the nucleotide from both strands of a double-stranded molecule. For exonuclease 3, all right, this one can only remove the nucleotide from the 3' end. So they can only remove the nucleotide one at a time from the 3' end. So these are the exonuclease. And then for the endonuclease, there are 3 different types of endonuclease. All right, so 3 different types of endonuclease. So one type of endonuclease can cleave the single strand DNA non-specifically. So like here we show you the S1 nuclease, as long as a single strand or a nick, and then they will just cleave it. It's just non-specific. As long as a single strand, they will cleave it. And then another type will cleave both single strand and double strand non-specifically. So like a DNS1. So in here you can see the DNS1, as long as a single strand, then they can degrade this whole single strand gradually, or double strand just degrade this completely. So you have these two which will cleave the DNA non-specifically, all right? And one only target to the single strand, and the other one, DNS1, target to both single strand and double strand. And then another type of endonuclease would be the restriction endonuclease, all right? And this is the most important one because they will target to a specific sequence and then cleave at a specific site. And this is also the most important one in the laboratory for gene cloning, because the restriction enzyme can specifically target to the target's nucleotide, and so that we can use this property to design the gene cloning project. All right, and then for the restriction endonuclease, all right, how do we discover this? All right, so basically this is the restriction endonuclease was discovered from the bacteria cells, and this is a defense system for the bacteria cell because as you know that the bacteria phage, they can infect the bacteria cell, and then they control the host system, and then later on they will lyse the bacteria cell in order to prevent the invasion from the phage. So the bacteria cell, they develop this defense system that can recognize the foreign DNA and then to digest the foreign DNA. So that's the restriction enzyme, how people discover, and how the restriction enzyme distinguish the foreign DNA and the host DNA. That's because the host DNA, after the replication, right, the DNA will be methylated. When the DNA is methylated, then the restriction enzyme will not cleave that DNA, and the foreign DNA, that's the viral DNA, that's not methylated, so that these enzymes, they will degrade those foreign DNA. So that's how the bacteria defense system differentiate the difference between the foreign DNA and the host DNA, and so they will just cleave the foreign DNA. All right, and there are 3 different types of the endonuclease. The type 1 is a one enzyme with different subunits for recognition, cleavage, and the methylation. And the recognition and cleavage site will be 1,000 base pair apart. Okay, and type 3, so one enzyme with 2 different subunits, one for recognition and one for cleavage. And the recognition and the cleavage is 24, 26 base pair away. So that means the recognition site and the cleavage site for both type 1 and type 3 will not be at the same location. Only the type 2 enzyme, all right, type 2 restriction enzyme, both the recognition site and target sequence will be at the same location, all right? And normally the restriction enzyme will recognize a symmetrical sequence, all right, and cleave at the recognition site. So here is the example of some enzyme. So for example, the ALU1, all right, they recognize AGCT, and then symmetrical. If you look at the bottom strand, it's also AGCT. So this is the target of ALU1, and ALU1 will recognize this sequence and cleave the phosphodiester bond between G and C, and that produce this blunt end, all right, blunt end DNA molecule. And then another type of enzyme like EcoR1, recognize these are hexonucleotide, all right, hexonucleotide. And most of the enzymes recognize the hexonucleotide, like the EcoR1. So these 6 nucleotides here, GAATTC, is the recognition site. And then if you look at the bottom strand and read it backward here, 5' to 3', also GAATTC, okay? So this is the recognition site, symmetrical. And then they will cut, the EcoR1 will cut at the location between the G and A here, and G and A. Therefore, you will cleave and then separate these 2 DNA molecules, so you get this sticky end, right? So this is blunt end, this is sticky end. And then for EcoR1, after you separate these 2 DNA molecules, then you produce this 5' overhand, okay, 5' overhand. Like here, it's the more detailed explanation. So basically, this one, cut here, the see the sugar phosphate backbone here. This recognize this AAGCTT. So the cut between the first A and second A, they cut here and cut here. All right. After they cut, all right, they separate 2 DNA molecules. See these 2 DNA molecule here, and then you have this 5' overhand, 5' overhand. Okay, and then for this PST1, so this is the in these three enzyme. This one is a PST1 enzyme. Okay, the PST1, they will cut at the number 5 location here, 5 and G here, 5 and G, A and G, A and G. So the recognition site is CTG CAG, right? So if you read backwards, CTG CAG. So they cut between A and G here, A and G here. So after they cut this phosphodiester bond, then you separate these two molecules. So you get this 3' overhand, 3' overhand. All right, so you have two different patterns. For the sticky end, 5' overhand, 3' overhand. It depends on the recognition site and also the cleavage location. So for this Hindi 3, we'll cut at between first and second nucleotide, and that give you 5' overhand. For the PST1, cut at number 5, number 6 nucleotide, number 5, number 6. So that give you these 3' overhand. And then for this small one, they will cut in the middle, and that give you a blunt end. All right? So that's the cleavage pattern for the restriction enzyme. And then the activity for the endonuclease. So basically the definition. Of course, the activity, the enzyme require the magnesium, right? And the activity is defined 1 unit, all right? Every 1 unit. That means you can digest 1 microgram. The amount of enzyme can digest 1 microgram of DNA in a total reaction volume of 50 microliter in 1 hour. So that's 1 unit. That's the definition of 1 unit. That the 1 unit of the enzyme can cut 1 microgram of DNA in the volume, reaction volume of 50 microliter at the in 1 hour. So that's the definition of 1 unit. And also there are also some different enzyme that can recognize and cut the DNA at the same site, but they are purified, isolated from different bacteria. And this kind of enzyme, we call it the isochisomal. That's the enzyme they recognize and cut at the same location, but purified from different bacteria. All right. So for example, the BSP1 from Bacillus and the ACC3 from the Acinetobacter, they basically will find and cut the same DNA sequence. So that we call these two enzymes are isochisomal. And some enzymes they can recognize and bind to the same sequence, but cut at a different position. All right. And then we call it the neochisomal. All right, they recognize the same sequence, but they cleave at a different location, and those enzymes we call neochisomal. Okay, and then also we have the enzyme called isocaldomel. Basically, they have different recognition site, but they produce the same nucleotide extension. Okay, so like NCO1 and PAG1. All right, they recognize different DNA sequence, but after the cleavage, they produce the same sticky end. All right, so that's the isocaldomel. So it's different enzyme. And the enzyme is so important. The restriction enzyme, the type 2 restriction enzyme is so important is because that we need this enzyme that can cut the DNA at a precise location, so that we can cut the vector and we can cut the insert, and then we can perform the ligation to ligate the insert DNA into the vector, so to perform the gene cloning. So that's why this is very important. And then this is normally how people perform the restriction digest in the laboratory. So it's in the microcentrifuge tube. And then you have this, you add all the reaction buffer and the DNA, and then you add the enzyme and leave 37 degrees for 1 hour. And then you can extract or you can perform the gel electrophoresis to examine the DNA the digestion pattern. Okay, and so this just will bring us to another topic that the enzyme can also be used, all right, to determine the fragment size, or even the sequence, or even the restriction location. Okay, so for example here, let's say this is a lambda DNA. We have 49 kilobase long, and then we have different enzyme, and then after the digestion, then we can predict how many recognition site of a particular enzyme in this genomic DNA. All right, and so to determine that, then you have to perform the gel electrophoresis. Okay, so as we all know, the DNA is a negative charge. So if we place the DNA in an electric field, and then we apply the electricity, and then the DNA will migrate from the negative charge, all right, to the positive charge. And if it's in the solution, all the DNA will migrate at the same speed, all right, if it's just the solution. And then you cannot distinguish the difference. However, if we introduce a network of complex of a pore, then the DNA will be, those DNA molecules will be restricted by their size, because if we introduce those pore for the network pore, then only the smaller DNA molecule can penetrate through the pore quickly than the large molecule. And so that we'll be able to distinguish the difference between the large DNA molecule and the small DNA molecule. Okay, the smallest will run faster, and so the distance of the travel will be the longest, and the large DNA molecule, okay, will travel slowest. Then you will have the shortest distance of the travel migration. Okay, so the agar gel electrophoresis, the migration distance is inversely proportional to the size of the DNA. Okay, it's very important. Large fragment migrate slowly, smaller fragment migrate faster. And of course, you have the tracking die to monitor the migration of the DNA. And then, so basically, the agar gel electrophoresis, because of the agarose gel, so we can separate the DNA based on the size. And then to visualize the DNA, we need the normally we use the astatine bromide, and then in the presence of UV light, then we will be able to visualize the DNA. Okay, and then the percentage of agarose used to make the gel can determine the ability of the gel to separate the DNA fragment of different size. Because if it's high percentage, that means the pore size is very small. And when the pore size is small, then that can help us to resolve the small size fragment. And then if the percentage is low, like 0.5%, then the pore size is large, then it will be difficult to resolve the small fragment. But it can help us to resolve the large size fragment. Okay. And to run the gel, we have to submerge in a buffer solution, and that can conduct electricity. And then we put the gel in the gel box, and then put the buffer, and introduce the electricity. Then the DNA can migrate. All right. And so this is the example to show you that. So this is the gel, and then you have well. A well, that's the place where you place your DNA sample, and then you have this electric field from negative to positive. After you load the sample, then you introduce the electricity, right, from the negative to positive. Then the DNA will migrate from negative to positive, all right. And then after a certain period of time, then the DNA, we can resolve the difference between the smaller DNA from the large DNA, because smaller DNA run faster than the large DNA. Okay, and this is the example of a DNA agarose gel, and here is a size marker. So we have 500 base pair DNA fragment, 1,000 and 1,500, 2,000, 3,000, 4,000. So you can see the smaller fragment run faster, and the large fragment runs slower. And also the resolution here, you can see for this distance, you resolve 500 base pair in this location. But the same distance, right? If you put it here, that'll be 2,000 and 4,000. That'll be 2,000 base pair. So basically, the resolution is better in the bottom here, and resolution is poor on the top of the gel. Okay, and the buffer. So these are two commonly used buffer, TBE and TAE. And TBE has a greater buffering capacity, give a sharper resolution than the TAE. Right, that's a TBE. And this is the standard gel box. All right, and then this is what, after you put the gel and then you set up the everything, then you can then put this into the power pack, and then to provide the electricity, and then to make the gel run, to make the DNA migrate. So this is the actual gel box that you can see. And when we perform the gel electrophoresis, okay, remember DNA is colorless. You will not be able to visualize the DNA. And then, so in order to monitor the migration, so you have to introduce the tracking dye. All right. So these are two commonly used tracking dye. All right. Bromophenol blue and xylene cyanol. All right. Bromophenol blue normally will indicate for 2% gel around 100 base pair DNA fragment location. For the xylene cyanol, will be about 800 base pair DNA fragment location. All right. So that from this dye, then you'll be able to monitor the gel electrophoresis. So the dye you can see here, just barely you can see the dye, right? That's the tracking dye, so that can tell you how far your DNA sample is. And then to detect the presence of DNA, so the cheapest way is acidity bromide, and acidity bromide can intercollect the double-stranded DNA. So under the UV light, then you will be able to see the DNA. Or you can use cyber green, cyber gold that can find a single-strand, double-stranded DNA. Okay. So and then they place the gel on the UV light, so you will be able to see the fragment of DNA. And this is just to show you the resolution of the actually the migration versus the size of DNA molecule, right? So here you can see the smaller size here, smaller size DNA, and the migration distance is longer. And then large size DNA, the migration distance is shorter. Okay, and this basically one important point here is if you run the unknown DNA fragment, all right, with the size marker, okay, and then based on the size markers DNA fragment and also their location, you can create a standard curve like this, and to show you the relationship between the migration distance versus the size of DNA. And then from this curve, then you can get the formula. From the formula, then you'll be able to estimate the size of a DNA fragment. Okay. So that's for example an example like this. So after you purify a DNA from an unknown microorganism, and you perform the digestion, and you run the DNA electrophoresis. So this is the unknown size DNA fragment, and here's the standard the DNA marker, so that you have the standard size here, and then from this migration distance, then you'll be able to create a graph like this. And after you create a graph like this, then you can estimate the size of the DNA fragment here. Okay, any question? Do you need me to go through this example to show you how to do this?
Yes, please.
Okay. Okay, I'll give you a quick demonstration, all right? Just a second here.
Oh, by the way, Professor, if you want to show us anything on the whiteboard, like I would recommend using like the PowerPoint on your computer, and then you could share the screen from there.
Say that again. Sorry, say that again.
So if you want to like show us something on the blackboard or like the whiteboard, so you could just like draw it on the PowerPoint or like any other like thing on like a blackboard or anything, and then you, instead of like doing it on the whiteboard and we can't see.
Yeah, draw on the blackboard. Yes, I understand, but it's difficult to use the mouse to draw anything. It'll be more convenient and also more precise to use the whiteboard to draw on the whiteboard, the actual whiteboard. Yeah, I understand you say I can do it on the blackboard, but the blackboard is really different. Even I use that the touchpad, use the finger or the mouse, it's really just difficult and ugly, and also not precise, and also take much longer time to do it. Okay, so now let's say for example in this example, right, for the gel electrophoresis, right, and now you have this size marker here, right? So you know the size of each of the fragment, and now you want to estimate the size of these three fragments. Okay, so what you need to do is just arbitrary. You need to have some measuring unit to measure the distance of each of fragment from the well, because that's the distance of migration. All right, distance of migration. Let's say, okay, so let's say you have to use the Excel, all right? So we have the fragment. Let's put the fragment, the size, the DNA size first. So it's a 564, right? And then 20, 27, 23, 22, 43, 61, 65, 557, 94, 16, 23, 1. So these are the non-DNA fragment size. And now you need to measure the distance, let's say the 564, right? Distance from this fragment to here. So let's say you use any ruler with any unit, doesn't matter, okay? So let's say the distance here is 25, let's say 25 centimeter. And then for 25, then this is let's say 9, okay, and then we have this 8 and 5, 3.5, 2, 0.4. All right, let's say this is the distance, all right? That the unit is arbitrary. You can use the centimeter, you can use the inch, anything, as long as you are consistent, all right? And then for these three fragments, you know that they say you measure the distance it travel. Okay, so number 3, they say it's travel 10 centimeter, all right? And then number 2, let's say it's a 6.5 centimeter. And then number 1, let's say it's a 4 centimeter. And then you want to estimate the size of these first 3 fragments. So first of all, you highlight these, right, and make a graph. So this is a graph, all right? So that's the migration distance versus the DNA size. Okay, so this is the graph. Okay, and now you right-click this, that show you this at the trend line. All right, at the trend line, and then you know it's not linear. It's a power. Okay, so it's a power, and also you want to know, display the equation, display the R square, all right? Then, so you see the equation here. So the R square is 94%, which is very close. And then this is equation. Let's make it bigger so you can see. So this is the equation, all right? So y equal to 14,559 multiply x to the power of a negative 0.891. All right, so that's the equation. So that's the equation. X is the migration distance. Y is the DNA size. So what you need to do is here. So you will do this. You will introduce the formula. 14,559 multiply x, which is this number x, okay, and then to the power of negative 0.891. And then so number 10, so for the number 3, all right, migrate 10 centimeter, all right. So then the size will be about 1871. And now you apply the same equation for the other 2, and so you get this estimate, all right. So you get the estimate of those 3 unknown fragments. Okay, any question about this? Do you all understand this procedure? Hadeo.
So Professor, it's just like finding the slope.
Finding the unknown DNA size.
But I don't understand. Like, how would you be able to find that? If
To
like
find that, so okay, you need to run your unknown DNA with the size marker. So size marker, I mean, it's the DNA that with the non-size, all right? And then after you run these unknown with the size marker, and because you know each of these DNA fragment, all right, so you can have these, you can create the graph that the size of DNA, and you have the migration distance versus the size of DNA. So that's the importance, and you need to have this size marker so that you can create this graph, and so that you can estimate the size of the DNA for the unknown. Okay, Tadeo.
Yeah, but then the R squared is equal to 0.947, right? So you
Yeah.
get that answer. So we're not gonna like square root this answer, or no? We're just gonna leave it
No, the
unchanged.
R squared just tell you how accurate this equation to predict the DNA size. So R squared, I mean 94.7% accuracy.
Oh,
And this
oh,
is the
okay,
equation. This is
okay,
the equation that you need. Okay.
okay. I get it now. Thank you so much,
Good.
Professor.
You are welcome. So that's the one application for a research enzyme that you can use that to predict the DNA size. Okay, so that's the one I just demonstrated. And now let's continue the research mapping the gene structure. Okay, so you cut the clone gene with research enzyme to pinpoint the location of the cutting site. And knowing the recession map is useful for making clones of small pieces of DNA, which is called subcloning. And then the small piece of DNA can then be sequenced. So the protocol of the recession mapping, so you digest the DNA with single or double recession enzyme, and then separate the DNA fragments through the agarose arrange fragment in order to make the map of recession site. Okay, so that's the protocol for recession mapping. So now, this is an example for the recession mapping. Okay, so and then you can determine the relative the location of the enzyme on the specific DNA fragment. So here they say we have the single double digestion for 3 different enzymes. Okay, so these are the 3 enzymes that we use on this in this example. So for XBA1 single digestion, you produce 2 fragments. So 2 fragments. Let's say this is a linear DNA fragment. If you use the XBA1 and then produce the 2 fragments, so that indicate there's only one cutting site for XBA1. And then for XHO1, also produce 2 fragments. That also indicate there is only one cutting site of the XHO1 on this DNA fragment. And for KPN1, it produced 3 fragments. So that suggests there are 2 KPN1 cutting sites. All right. And here's the size after they cut. All right. And then this is double digestion. If you perform the double digestion, then you produce these 3 fragments, and this is size 3 fragment. This double digestion 4 fragment, and these are the size. Okay. And now, from this information, then we have to interpret the enzyme location on this particular DNA fragment. Okay, to do this, then I have to turn on the video. I'm going to do it. I'm going to do the demonstration on the whiteboard. Okay, so that you know how to do the recession mapping, which is also important. Okay, and the recession mapping will also be on the exam as well. Okay. So I'm going to do the demonstration, how to calculate, how to do the recession mapping. So you have to turn on the video, okay? So now, the recession mapping, so for XBA1, all right? So you have 2 fragments, and then you have 24 and 24.5 kilobase, all right? And then XHO1, also 2 fragments. And then you have 15 and 35.5, or 33.5. Okay, 33.5. All right, and then for the double digestion, XBA1, XHO1, all right? Blurry. So you need to, let me move my camera closer. Or maybe I should not share the content now, okay? I'll stop sharing maybe. Okay? Is this better now? If I stop sharing. Okay, so now let's determine these two enzyme location first, all right? So we do the single digestion, and double digestion. For this one, that produces 3 fragments, and the 3 fragments are 9 kV, 15, and 24.5, 3 fragments. Okay, so to determine this relative location, first of all, you can choose any single digestion, right? So, and then go to the double digestion, and finally you can confirm with another single digestion. So, first of all, you make a table like this, and then you label the step that you're going to analyze. All right. So you're going to do this number 1, and then you do double digestion number 2, and then number 3 another single digestion. All right. So let's look at number 1, XBA1. XBA1 give you 2 fragments, and then that suggests linear DNA, 2 fragments, so that means there's only 1 cut site. After you cut here, then you produce 2 fragments, all right? So you only have 1 cut site. And then what's the location of XBA1? One is 24, the other one is 24.5. So that means that'll be in the location somewhere in the center here, all right? XBA1, all right? And we'll cut here so that you can have 24 kV and 24.5 kV. Okay? So that's first step. So now you put XBA1 in the location in the DNA now, because that gives you 24, 24.5. And now the step 2, look at the double digestion. Like 3 fragments, 9, 15, 24.5. So immediately when I see this number, so I know this 24.5 is here. All right, 24.5 is here. And these two, 9, 15, that give you 24 here. Okay, so that means this is XBA1. So that means the XHO1 cutting site must be in this location, in this fragment here. All right, must be in this fragment here. All right, it's not here because this fragment is intact, still 24.5 after you perform the double digestion. So the XHO1 must cut this fragment, and so that gives you 915. All right, so after the digestion, you get 9 and 15. So that means the location of the XHO1, it could be here, so that you can produce 915, or it could be here, so you get 15, 9. All right. So you can have 2 different possibilities. All right, two different possibilities for the location of XHO1, because you can produce 9 from here, 15 from here, or 15 from here, 9 from here. All right, so you have two possible locations. So to finalize the XHO1 location, then this is step 3. The step 3 tell you the XHO1, when you cut it, you produce 15 and 33.5. So that would just immediately eliminate this possibility, because only the XHO1 at this location will give you 15 kV here, all right? And then this part will be 9 plus 24.5, 33.5. All right, so XHO1 must be here, all right? And you have 15 kilobase here, and here's 9, and here's 24.5. Okay, so that's how you determine the restriction mapping. And now you finish the SBA1, SHO1, then we can look at the KPN1. Okay, so for KPN1, the KPN1, the KPN1 3 fragment, all right? KPN1 3 fragment, 1.5, 17, and 30, right? And then the double digestion, SBA1, KPN1, it's a 4 fragment, okay? And then there'll be 1.5, 6, 17, 24, okay? So again, you assign the number, all right? So XBA1, you will look at it first, okay? XBA1, you look at it first, 1, all right? And then you look at the double digestion, 2, and finally 3, KPN1, all right? So now from XBA1, okay, XBA1, again, you produce 24, 24.5, all right? That's XBA1. And now look at the double digestion. Double digestion again. You see the 24 here. So you immediately identify this 24 is this 24, okay? And so that means these 3 fragments will come from 24.5, because you add up the total, that give you 24.5. So that will suggest that KPN1 cutting side would be from this fragment, okay? And now there will be different possible combination of this number. So this number could be, let me erase this. So XBA1, all right, and 24 kV, and then the KPN1 must be here, 24.5 kV, all right, and then we have 3 fragments, all right. The 3 fragments can be 15, the arrangement, all right, the different possibility, all right. So it can be 15, 6, 17, 15, 17, 6, 6, 15, no, it's not 15, 1.5, 1.5, sorry, 1.5, and 17, and 6, 17, 1.5, all right, and then 17, 1.5, 6, 17, 6, 1.5. So you have these 6 possibility, All right, these 6 different arrangement. And now to determine which one is the correct one, you have to look at the single digestion, the step 3 here. All right, so after the single digestion of KPN1, then you get 3 fragments, 30, 20, 17, 1.5. And remember, because here is 24, all right, 24. So you need to have a 30, all right, 30. KPN1 doesn't cut here. So you need to have 30. So immediately you know you need to have a 6. You need to have a 6 here. All right, so that 6 here, so that can give you 30, kilobase long. Therefore, you eliminate this. All right, and these 2 will be your answer, because that should be possible cutting site for the KPN1. It could be here, or you have a 6, 1.5, 17, all right, or 6, 17, 1.5, all right, so that's KPN1 side. Okay, any question? So that's the restriction mapping, which is also very important. And so in this graph, it shows you that the poor resolution of a DNA molecule above a certain size in this range, and this range give you a more adequate resolution. So that means the smaller size of DNA, all right, they can travel a longer distance. They give you a better resolution. And large size of DNA fragment, they give you a shorter travel distance, and then so the resolution is low. So this is just like the actual DNA gel I show you here. So let's go back to that gel. So like here. So as I just mentioned, this here, for this distance, all right, this distance that resolve 500 base pair, but for the same migration distance, this long distance, if you bring it back here, you see the same distance, but this in here they can only resolve 2,000 base pair. All right, between 2,000 and 4,000 in this range is 2,000, but in this range you can resolve 500. Therefore, you have a better resolution in the lower half of the gel than the upper half. So that's why we have this curve to show you have better resolution in this range and poor resolution in this range. Okay. And then the ligase. Okay, so the ligase. So basically, for during the gene cloning, you cut the DNA, and then you have to ligate the insert into the vector. So for this, you need the enzyme called ligase, because it can repair the phosphodiester bond. All right, they can repair the single strand or the doubles to join the 2 nucleic DNA molecule. Okay, so you need the DNA ligase. So you can ligate this blunt end. These are 2 different DNA molecule. Then you can ligate together, but it's not very efficient because you have to repair the, you have to ligate this. You have to repair this phosphodiester bond. Right, you have to use a very high DNA concentration so that you can increase the chances, right? And then for the sticky end, very efficient, because they say these are 2 different fragments, and then you have sticky end, 5' sticky end. Then if they complementary to each other, they can form the hydrogen bond first. Therefore, they are stable, so that you can fix this single-stranded phosphodiester bond. All right, so that's why it's very efficient. And this is just to show you the ligation. We need a ligase to insert a piece of DNA molecule into the vector, right? And then during this process, if it's a blunted end, then the efficiency is low. Then we can fix this by putting a sticky end to the blunted end molecule, all right? So first method is the linker, all right? The linker. So basically, you link a short piece of double-stranded DNA that contains a specific restriction site. So let's say we have a typical linker like this, all right? And the linker is 14 nucleotide long, and inside contain the BNH1 site, BNH1 cutting site. All right, so you mix the blunted end molecule with high concentration of the linker. All right, so in the end, this linker will bind to the blunted end of the molecule. After that, in the presence of ligase, of course, then you perform the BNH1 digestion, and then that will give you this sticky end. All right, then this DNA molecule will be easy to clone into another BNH1 cutting fragment. So that's the linker. So you can fix the problem if it's a blunted end, and to increase the ligation efficiency. And the second method will be adding the adapter. All right, so the adapter will be a shorter synthetic oligonucleotide, and that oligonucleotide has a sticky end for a particular enzyme. In this case, they show you the BNH1 sticky end. All right, BNH1 sticky end, and then you attach, you mix. They could adapt, of course, they could ligate to each other, and then you mix this adapter with the DNA molecule. It's blunted end DNA molecule, and then after that, then you can cut the adapter, okay, through the BNH1 and to give you the sticky end. So that's adapter. So linker, adapter, and then another method would be another method is this the homopolymer tailing. Homopolymer tailing. So basically, you have this is the blunted end. Then you synthesize the homopolymer tail by adding the terminal transferase plus the specific nucleotide you want to add. So in this case, they show you the deoxy-CTP, all right, in the presence terminal transferase. Then they will add the C in the end of the 3' end, so that you get the poly C tail. And then you can have a poly G tail for your insert. And then you can ligate these two molecules together. All right, so that's the homopolymer tailing. And polymerase. So basically, it's very commonly used in the PCR reaction because the DNA polymerase is responsible for the DNA synthesis. All right. And the 3 different DNA polymerase that make the DNA in E. coli. And the 4 commonly used DNA polymerase in the lab. We have DNA polymerase 1, and clinal, reverse transcriptase, and Taq DNA polymerase. DNA polymerase 1, that's a regular polymerase that can synthesize a DNA fragment. Okay, so like this, the DNA polymerase 1, if it's a nick, then they can synthesize, and also they replace the existing nucleotide, keep bringing the new synthesize new DNA strand. And the clinal fragment is only a part of the DNA polymerase 1, and this clinal does not have an exonuclease activity, therefore they can only fill in the gap. They cannot replace the existing nucleotide. Okay, that's the clinal. And reverse transcriptase, basically they use the RNA as a template to synthesize the complementary DNA. And Taq DNA polymerase. So this is the important one because this is the one that can perform the function during the PCR reaction, because the PCR reaction require the high temperature during the PCR reaction, 94 degrees, and the enzyme, these polymerase are enzyme, they are protein, normally they will be denatured and lose the function. However, the Taq DNA polymerase can survive at 94 degrees, so the Taq DNA polymerase is particularly useful in PCR reaction. Okay. In polymerase 1, this is the exonuclease activity. All right, so that's why 5' to 3' exonuclease activity. That's why the DNA polymerase 1, they can degrade the existing nucleotide and then replace with the synthesized nucleotide. Okay, that 5' to 3' exonuclease. And DNA modifying enzyme, so that's topoisomerase. And the important one is the gyrus. In the bacteria, is a topoisomerase 2 because they can cut, they locate in front of the replication fork, so they can release the tension during the replication. That's a DNA modifying enzyme. Or the alkaline phosphatase, right? That can remove the phosphate group at the 5 terminals of DNA molecule. Okay, so we have this polynucleotide kinase at the phosphate group to the 3,5' terminal, or the terminal deoxynucleotidotransferase. You can add the nucleotide to the 3' end, just like I show you the polymer tail. All right, so you use the terminal deoxynucleotidotransferase to add the specific nucleotide to the 3' end. So these are the DNA modifying enzyme. So DNA modifying enzyme like this, the alkaline phosphatase. All right, so 5' phosphatase will be removed, replaced by the hydroxy group. That's alkaline phosphatase. And this is important during the gene cloning, because when you use alkaline phosphatase, you can prevent the vector self-ligation. All right, if you use alkaline phosphatase to treat your vector, then they replace the phosphate group with the hydroxy group, and then the vector will not be able to self-ligate. And the PNK, the polynucleotide kinase, basically they will attach the phosphate group to the 5' end, and in this case they replace the hydroxy group. Okay, from here, replace the hydroxy group with the phosphate group. Okay, and then this terminal deoxynucleotidotransferase add the nucleotide to the 3' end. So that's basically today's lecture. Okay.